Hello, my name is Joey Peters, and my project is regulation of the BMP receptor WIT in Drosophila melanogaster. So our lab studies neuronal development in fruit flies or Drosophila. We specifically study this at the neuromuscular junction. This is the region where a motor neuron innervates a skeletal muscle cell. The amount of synaptic growth or lack thereof can be quantified by counting the number of terminal axons. My project involves studying a specific gene called WIT that we believe acts in a signaling pathway responsible for neurodevelopment. My hypothesis is that tricornered, a gene previously identified by our lab, phosphorylates WIT to inhibit synaptic overgrowth. To test this, we needed to make two mutated WIT constructs, one that acted as always phosphorylated and one that acted as never phosphorylated. To do this, we had to put an ATP scar into the gene. The way we put an ATP scar into the gene was through a process called CRISPR-Cas9 technology. CRISPR-Cas9 is essentially a cut and paste mechanism. We synthesize a guide RNA that complexes with the Cas9 enzyme and tells it exactly where to cut in the fly's DNA. Then in a process called homology-directed repair, a piece of DNA that we make, called the homology-directed repair template, is pasted into the cut. This piece of DNA is what contains the ATP scar. So in order to do CRISPR-Cas9, we had to make our guide RNA and we had to make our homology-directed repair template. To generate the HDR template, 4 distinct DNA segments were amplified by PCR: a left WIT flank, a right WIT flank, a plasmid backbone, and an insert containing DS-Red, ampicillin resistance, and the ATP scar. We will get to DS-Red later. These were then confirmed by gel electrophoresis, and then using specific primers that we designed, their overlapping ends annealed and were ligated in an assembly reaction. Then the HDR template was transformed into E. coli for replication. To generate the guide RNA, we used a site-directed mutagenesis reaction. This generated a small substitution in a previously synthesized plasmid that was homologous to the anticipated cut site in the WIT region, essentially telling Cas9 exactly where to cut. The guide RNA and homology-directed repair template were then analyzed by sequencing. A chromatogram is pictured on the top right that visually represents confirmed base pairs of each construct. By cross-referencing these to the published Drosophila genome, we ensured that no spontaneous mutations occurred. After confirming this, both the guide RNA and homology-directed repair template were injected into a fly that expressed Cas9. To ensure that CRISPR-mediated homology-directed repair occurred, the DS-Red selector was used. This is not normally expressed in flies and generates red fluorescent-eyed flies, thus allowing us to visually select which flies underwent CRISPR-mediated homology-directed repair and which did not. So the future steps involve generating yet another plasmid, called the ATB plasmid, that will be placed into the WIT gene at the ATP scar. This will generate the always and non-phosphorylatable WIT constructs. This will then allow us to count the number of boutons in these mutants and determine if significant synaptic overgrowth is present, with or without tricornered. I'd like to acknowledge the Collar and Weaver Summer Research Fund, the John Carroll University Biology Department, the Breuer Lab at Case Western Reserve University, National Science Foundation, and Caitlin Totoret. Thank you.